जी सर। हाँ जी सर, स्टार्ट करें गूगल मीट। गूगल, हाँ जी गूगल मीट पे आ गए सर। जी सर। और जी सर। ओके सर। So, very good evening, class. Very warm welcome to the second day of the Proteomics classes. Uh, Please unmute yourself. Sorry, mute yourself and keep your cameras off. It's not required. Soon I will share my screen. I will start slowly and slowly. Please share your queries on the right hand side in the chatting box, whatever the queries that you have. I will be happy to solve them. So today I am back from the trip outside Delhi. So today we will not face the problems that we were facing yesterday. And but one thing that I have to explain is that so we are working on the Google Meet, so we don't have option of recording on Google Meet. But I'm trying to record these on the YouTube also. So later, if things goes fine, you can have the session from the YouTube also. But if it is stopped in between the streaming, so I cannot help. So we might lose the things because I'm doing two things at the moment: Google Meet and live streaming. And yeah, so that's why I was preferring. That was I was preferring YouTube over Zoom or Google Meet because while I was presenting, some more students will be leaving the sessions. So when they come, so I have to come and open the window to let them in. So that's really irritating and very disturbing also. So there were some students who were like creating havoc. I would use that word and that oh we want this, we want that. If this not happen, like creating such a big mess for everyone. So please, uh, no bache. So we have to buy something uh, Google Meet subscription to record the session. So I don't see any options of recording it. So YouTube sessions are fine for us. Like yesterday, I had a problem with the networking, but today there's no issue. I'm back at home. So I had just finished 2 hours of MBBT class where I didn't have any problem uh, and nothing got stopped in between. So everything went very smooth. So YouTube was fine. So everyone is saying YouTube is fine. YouTube is fine. Yeah, Google Meet, I also don't like it. So I just came because of some students were screaming on the WhatsApp, making, what should I say, हमारी calendar खाना दो, calendar खाना दो, like that. So please maintain decor, be a nice students, humble students. Yeah, I know that. The recording is, I'm not sure. I put in up the recordings for you, but it could stop because it's telling me warning. So recordings could be stopped at any time. So one more thing is that if I have been continuing with the lectures, so if I'm presenting something, so some students will be in the line to attend it, to join it. So they can only join if I share my screen, if I click on the screen. So maybe I should start sharing the screen now. So this is my screen and somehow we have to buy subscription to record the Google sessions. So we don't have that. So this is free session. So if you go there, attachments, so I don't have any options of recording in the Google Meet. Yeah, YouTube will be more convenient. I also agree. But you are all joined now, 72 students. What can I say? So the YouTube link is also on. If you want to switch from Google Meet to there, you can do it now also. I can share the link here, but I have to stop. I have to stop. I have to stop Google Meet then. So you're all ready to switch to YouTube then. Okay, fine, fine. So this is the link for day 2, Proteomics, Edufabrica, Simranjit Singh. So both are open. So I would recommend to students, let's continue with the Google Meet. Don't leave this part. Don't leave, don't leave, don't leave. So you already started to leave. Okay. So students at Google Meet, please, I have shared one link on the WhatsApp. Just click on that and come over YouTube so that we can have recorded session also. Yeah, just leave these sessions here and check your WhatsApp group. Yes. We will create a telegram group for all of you, or maybe later, so where you can pin your documents. And please talk to Vatsal sir and Shivani ma'am about that. For the moment, please shift to YouTube so that we can start session. It's already 30 students have been joined there, so please join it at the WhatsApp group. It's on the WhatsApp group, please. Yeah, you can go ahead for the 15 or 5 groups people maximum or 7, and go ahead with that. So it's no problem. Hello, sir. Oh, hello, students on YouTube. I hope things are clear. So most of the students, please leave this part and join the link, or you can join it here also on the YouTube. Please come on the YouTube streaming so that we can have recorded version. Yeah, Vatsal sir. We will continue on the YouTube. and we will share in the WhatsApp group the documents. No worries. So already 50 students have joined there. Oh yes, I was driving today whole day and I came. So 11 students left. Please left it and join it with the above link on the YouTube streaming. Sorry for creating a bit chaos, but let's maintain the decor as it is. Okay, I will stop this meeting now. I hope everyone has understood the point. We are working from YouTube streaming. So I will close this. I hope everyone is conveyed with the message. So 8 people who are there, please join the link and come to the YouTube streaming. Okay, it's already started. So sorry students, I couldn't provide you today the topic for proteomics, but you can look for it today. Whatever the topics that you think that is suitable for the review writing, you can go ahead with that. But the rest of the topics I will surely share with you in upcoming time. So don't worry about it. Uh, so let's start for today's session. I hope most of the things are clear with you, like assignments, review writing. Just a second. So uh, let's start the session. So today we will be dealing with the protein. So somebody was asking yesterday, uh, is it assignment as a PPT or Word document? As a Word document or PDF, whatever you feel, just try with that. My internal microphone. Let me check. So, students, we should start now. I think we had enough talk. Um, so the disturbance sound is coming from my it's from my laptop, which will be gone soon. So don't worry about it. So assignment is not necessary. It's not compulsory, but if you write it down, it's quite good for us. And and so don't worry. So we're gonna study about today about protein stability and folding, how proteins are folded and how the stability is maintained. We will deal with that part for today. So learning outcomes that you will see from these lectures from today is that you will be acquainted with the basics of protein structure, including structure of amino acids, its overall effect on protein folding. You would be understood about few important principles behind protein folding and mechanistic hypothesis governing the protein folding. And further, you will understand the concept of protein stability and mechanism underlying the stability of your proteins. So that will be the learning outcome for today. So protein introduction and evolution. So proteins are linear polymers built of monomers, units called amino acids. Proteins contains a wide range of functional groups. Functional group includes like alcohol, thiols, thioethers, carboxylic acids, carboxamides, and variety of basics groups. So further amino acid structure we have discussed yesterday. It's looks like this: NH, COH, R side chain, and H. So this is the basics amino acid structure. So they are essential amino acids and non-essential amino acids. Essential amino acids are one that cannot be produced by the body. So result, we need them from our food. So there are 9 types, that is histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Whereas the non-essential amino acids includes humans that is not essential to the human diet, and there are 11 non-essential amino acids, that is alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acids, glutamine, glycine, proline, serine, and tyrosine. And further, peptide bonds, there are 2 or more amino acids are joined by condensation reactions, that is removal of water from the peptide bonds. So this is how your C1 and N2 they make together your peptide bonds with oxygen and hydrogen to be on the side chains. So particle double bond vector is either strengthened or weakened depending upon the environment that it is in. It is shorter than a single bond and rigid and planar. Proteins can interact with one another and other biological molecules to form complex assemblies. Assembly includes macromolecular machines that carries out the accurate replications of DNA, the transmission of signals within the cells, and many other essential processes. So how evolution is done? So according to evolution, Charles Darwin first established evolution by common descent as the dominant scientific explanations of biological diversity. So the hedgehog has no pigmentations due to mutations, and mutations are permanent, transmissible changes to the genetic material of cell or viruses. And these mutations results from the errors in the DNA replications during cell divisions and by exposure to the radiation. So basic principle behind it is that protein evolution is based on the study of two patient genetics and evolutionary biology. If the protein accumulates a destabilizing its ability to function, natural selection will rapidly remove it, and protein duplications will happen further. Recombination is a process that results in genetic exchange between the chromosomes or chromosomal regions, and gene convergence is a type of recombination that is product of DNA repair where nucleotide damage is corrected using an homologous genomic regions as a template. And drift is the change of allelic frequencies from one generation to next due to the stochastic effects of random sampling in the finite populations. So we are just talking about how protein and evolution they coexist, how various what do you say, recombinations, evolutionary and population genetics, Charles Darwin theory, mutations, they all belong to the protein that we are having at the moment. How everything has been staged, how everything has been produced. So genetic drift is the change of allelic frequencies from the genetic generation to next generation due to stochastic effects of the random sampling in the finite populations. So rate of evolution, histone H4 shows extreme conservation. It has essentially the same sequence in all eukaryotic examined. So histone 4 remains same. Its functions are extremely dependent on the entire structure of it. So level of protein organizations. So they are primary, secondary, tertiary. So we will deal with this part in this topic. The sequence of amino acids in the polypeptide chain is known as primary structure. In the secondary structure, you have formation of alpha helix and beta pleated shades due to the hydrogen bonds between the peptide backbone. Then folding of helix and sheets influenced by the R group folding. And in the potentially structure, quaternary structure, the association of more than one polypeptide into protein complex influenced by the R group bonding. So this is how your four protein structures are made. So let's see their figures. So primary structures looks like this. In the where primary structure determines the folding of molecule. So here you can see glycine, isoleucine, valine, cysteine, glycine, glutamate, alanine, serine. They are being joined by a disulfide bonds. Cysteine and cysteines are making disulfide bonds. This is a primary structure. So peptide bonds are being made. Two or more amino acids are joined by condensation. Removal of water forms the peptide bonds. The partial double bond character of the peptide bond, thus the conformation a polypeptide chain may assume, it is shorter than the single bond, rigid and planar. The primary sequence must organize itself to form a compact structure. This is done in an elegant fashion of forming its secondary structure elements. The peptide backbone has areas of positive charge and negative charge. These areas can interact with one another to form hydrogen bonds. The results of these hydrogen bonds are two types of structures, that is alpha helix and beta braided sheet, as we have discussed before. So alpha helix are the most common conformation. So alpha helix structures are the most common conformations. It is spiral structure and tightly packed coiled polypeptide backbones with extending side chains. Yeah, and R group protrude outwards from the helical backbone. So height of single turn of helix is 5.4 angstrom with 3.6 amino acids residues, whereas the right-handed alpha helix predominates in nature. It is stabilized by the H bonding between the amide hydrogens and carbonyl oxygens of peptide bonds. So this is how your alpha helix looks like. So you have these gray as a carbon, white as a hydrogen, oxygen, nitrogen, and an R group. So this is how carboxyl terminus is being arranged. And in between each residue, there is a size of difference of 5.4 angstrom. That is between 3.6 residues are included in between them. So that's how your alpha helix is made, alpha helix pleated sheet. And then beta pleated sheet is the backbone of polypeptide chain is extended into zigzag rather than helical structure. So earlier we saw helical structure like a DNA, but in beta pleated sheet, it's more uh, like this one. Whereas helix structure was like this. It was like helix. Zigzag polypeptide chains can be arranged side by side. So there's a zigzag option like this, by hydrogen bonds between adjacent segments. And the adjacent sheets can either be parallel or anti-parallel. So beta sheets looks like this. So as I showed you, there is a zigzag fashion made. They are anti-parallel to each other, and there is also parallel to each other. So this is how beta pleated shades are being made with a chain of various amino acids and polypeptide chains. Folding of helices and sheets influenced by R group bonding classified as fibrous proteins and globular proteins. Fibrous proteins chains arranged to form long strands, usually that consist of a single type of secondary structures and structures that provide support, shape, external protections to vertebrates are made up of fibrous proteins. Whereas the globular proteins, chains arranged to form spherical shapes and often contain several types of secondary structures. So enzymes are mostly the globular in nature. So there are two types of folding. According to that, tertiary structure could be fibrous and globular proteins. So this is alpha keratin example is there, keratin alpha helix with the two chain, and protofilaments are being made around a length of 20 to 30 angstrom of the diameter, and the protofibril is made like this in this case. And this is a cross section of hair. So alpha keratin is our hairs. So we having our cell structures here. Intermediate filaments are present. Protofibril are present here. Protofilaments are present. Two chain coiled alpha helix structures are being present inside of that. So you can see how within the cell two chain coiled uh, alpha helix structure is being present in the cross section of hair. Somehow it could be, can say, an analogy of your nucleus having a DNA inside that in the alpha helix structure in the presence of your inside the chromosomes, and inside the chromosomes around that uh, histones it is covered with alpha helix sheets. So the same way your proteins alpha keratins are also present inside your cells in the form of protofibrils and protofilaments, and then there is a two chain coiled alpha helix chain is like this. So this is the structure of collagen. This is also alpha helix structure. So you can see very nicely it's coiled together. This is further its advanced versions of it with more structures coming into it. And then in the tertiary structure, that was about alpha and beta. In the tertiary structures, factors influencing the tertiary structures includes hydrophobic interactions. Oh, sorry, we were discussing about tertiary structures so far. So factors influencing the tertiary structures includes hydrophobic interactions, that is with the water heating interactions, and then hydrogen bonding, disulfide bridges, ionic bonds. So all these are applicable in the case of tertiary structure. Then comes the quaternary structure, that is the fourth one, which results from the interaction of independent polypeptide chains. Factors influencing the quaternary structures includes hydrophobic interactions, hydrogen bonding, the shape and the charge distribution of amino acids of associating polypeptides. So these are the various factors in the quaternary structures. They are being factored. And further, hemoglobin is made up of four polypeptide chains. So this is the quaternary structure of hemoglobin shown with the dimers formation also, with having a heme group present in there on four sides. So now the next topic, we are done with the various structures. Now we have discussed so far about various alpha, beta, tertiary, primary, secondary structure, tertiary structure, quaternary structures, how alpha structures, secondary is divided into alpha and beta, how tertiary structures have helices and sheets influenced by the R group bonding, and we have discussed in the quaternary structure that it's made up of all the things that is in the primary, secondary, and tertiary, and made them together in the quaternary structure. So that was the basics level of protein organization. Uh, we have discussed so far, and before that we have discussed about the evolution of protein, how protein has been evoluted from the point of view of Charles Darwin, then evolutionary biology, and how population genetics also plays role in the evolution of protein stability and folding. Now, after doing all these topics, now comes the topic of uh, protein stability. How protein is stable? What are the factors that are being taken care, and what are the things that will be uh, how protein will be stable on basis of which factors. So protein stability is a net balance of forces which determines whether a protein will be its native folded conformation or denatured state. So there are two states of a protein. That is, one is a native state, that is they are folded, and one is denatured state in which they are unfolded state. They are open without any bonds, without any secondary or tertiary structures, and they're mainly present in the primary structures. So these all these structures of what we have discussed so far, they are mainly dependent upon the forces, the balances of the forces that are present on the protein. And protein stability normally refers to the physical stability, not the chemical stability. So we mainly check the physical aspect of stability here, not the chemical, like mainly the thermodynamics. So all the energies that maintain protein stability to be as the main primary goal. So what are the forces that are involved in protein stabilizations? That's our hydrogen bonding, van der Waals interactions, ionic strength, disulfide bonds, hydrophobicity, that is the dominant force in protein folding forces involved in protein stabilization. So out of these five forces, that is hydrogen bonding, that you might aware with the hydrogen-hydrogen, van der Waals interactions that are happening because of the weak interactions of the bonding, ionic strength, disulfide bonds, and hydrophobicity. And out of all these five, hydrophobicity is a dominant force which hold the protein forces in the protein stabilization. It's maintaining the protein stabilization in that case. Now comes the factors which are affecting the protein stability. Its temperature. So extreme temperature could make protein quite unstable, not good for the work, for the experiments or for the functions that they are doing. pH, extreme pH also cause instability to the protein. If there is extreme pH, that could also lead to the extremely bad functioning of your proteins. Organic solvents are also the reason unstable to protein. Urea, like these are the chaotropic agents like urea, guanidine, hydrochloride, which destroy the tertiary structures. So mainly they are destroying the tertiary structures, making them unstable, and not able to working out with that. If within these secondary structures, tertiary structures, the disulfide bonds are broken and then carboxymethylated with idioacetate. Yeah, if the disulfide is broken, that is reduced, then they are carboxymethylated with the idioacetate. The resulting protein is denatured, that is unfolded and mostly unfolded. So if you just broke the disulfide bridges and then you adding idioestimate ERR, then IAA in English, in German we say ERR, the resulting protein that will be producing out of it uh, will be the denatured or unfolded proteins. So ligand binding so increases the stability of the proteins at the end. Ligand binding increases the stability of the proteins. So on that note, if you have any questions, you can feel free to ask me now. I will take a 5 minutes break from now onwards just to get a sip of water. So it's 35. So we meet at 40 then. But chirotrophic agents are the one that we have discussed just now with the urea and guanidine hydrochloride, which destroy your tertiary structures. they are the character. So can you please explain content other than the slides too? Yeah, yeah students, today we are having uh, lectures basically uh, that we have taught at the college level also. So this is how the system is in the college level. I don't know if you have these lecture based things. In Germany we were taught like this only. So they are lecture based, they are slides, and we are taught on the basis of that. And these slides are made in very from reading a lot of books, and a lot of work has been done to do so. So these slides are not just made like a single day. So it takes a lot of efforts and a lot of thinking to bring the summary of all the that you should not have to read books. But today's problem with the students is that they want everything, and they want things to be very concise, and they want eatable in the just front of the desk, and they don't think that how things are being created for them after having a long process. So please understand the pain behind the making these slides. They were not just made in a single day. So these slides are made with lot of time. So you're all young blood. I can understand that, and you will not understand how we are working for you. So बच्चे mass M stands for mass, Z stands for charge. So basically we check mass by charge ratio while doing so in the mass spectrometry. That's the main principle behind it. So, बच्चे, the questions that I will read after the session is over, or in between the sessions before the break. So that's why I came back and to read your questions. But I have to admit, I had very nice students since July. I'm teaching students, so no one was complaining us in that high level. So please, rather than to be a problem creator, be a problem solver. You are all students, and you can find solutions between each other. You are here to learn, and we will provide you the best things that we have. And please trust us. That the trust issues is the problem. Uh, that things are not working as it is or something. So please maintain the decor of the workshop. So students, just give me 5 minutes. Just drink some water. Then we start with further topics. Thank you very much. Um, I join you soon. So students, uh, so the faltus, your questions are interesting. Please write me down personally, so I will reply to you. Yes, students. I think PBS would be a good solution. Okay, yeah, might be because your palette is not being dissolved, right? So that's the problem. Sir, I'm doing protein. Just students, listen to this question. I'm doing protein profiling research work. So first, doing extraction of protein by TCA method, then acetone method, then phosphate TC acetone method. Okay. End of the each protocol palette apron is not dissolving any buffer, even not in dehydration buffer. Yeah, try PBS. Maybe that might solve your solution problem. So let's continue. Let's make sessions more interactive. Uh, if I'm not able to answer, if somebody knows the answer, please try to answer that. So it's like a family. We all work together, not just to working on the ego issues. So I can see some some ego issues are being being present here. So try to dissolve your ego and try to start thinking from other perspective that we are here for learning, whatever the way is. Because in earlier time we were taught on the blackboards. Teachers comes and they just give lectures and they go. And here you are being provided with proper presentations, with each things been worked for 1 month, and a lot of effort has been done while doing so. So please respect to that. So I will continue. So we were at the disulfide bonds. So now comes the protein denaturation. So there's a loss of three-dimensional structure sufficient to cause loss of functions is called denaturations. And alternations in the environment such as pH, salt concentrations, temperature, which disrupt bonds and forces of attractions, are the ones that deal with the protein denaturations. The protein folding, a folding protein follows multiple pathways from high energy and high entropy to low energy to energy and low entropy. So protein misfolding, so disease, amyloid diseases results from the protein misfolding. So they are not being folded in the right way, so they are folded in other way. So that leads to the amyloid disease. So then there is a paradox called a Leavenworth paradox. So in this, all folding protein molecules are guided by an energy bias to transfer an energy landscape towards the native conformations. Energy landscape, your protein finding. So like this is a linear amino acid strain. Then they say that the paradox, Leavenworth paradox, like this, bumps into the funnel present energy barriers, and folding will take path the least resistance and its final state. So least energy resistance it will take from the final state of 3D conformation. Factors of protein misfolding. So the disease that we saw, that like amyloid disease happened. So there's proteins are misfolded. Somebody also asked these questions before. So answer to that. So absence of normal supporting cofactors. So absence of chaperone proteins, change in temperature and pH. So all these things are the factors that dealing with it. And also mutations, premature termination of translations, fault in the post-translational modifications, strong promoters, high inducer concentrations, reasons for protein misfolding, loss of conformation due to stress. So chaperones, what are the chaperones? These are the protein molecules which help in the protein folding. They help in the protein folding and prevent the aggregations. So examples are GROEL as a bacterial chaperones and HSP heat shock proteins in the eukaryotes. So these are the various chaperones which help in the protein folding and misfolding. So this is the various chaperones looks like. This is chaperone in, fully assembled, cap for a hollow cylinder. Then this is an unfolded polypeptide enter the cylinder from one end, polypeptide chain, and this cap attached to the end of the cylinder, causing the cylinder to change the shape, and it's create a hydrophilic environment for the folding of the polypeptide chain. And there's a correctly folded proteins here. The cap comes off and the properly folded protein is released. So like this, the chaperones are acting like a bag. So where polypeptides goes in, and then they goes out, being a properly folded, correctly folded proteins. So they have a cap and hollow cylinder. So together with that, they are doing act of uh, an act of uh, yours folding of the proteins as the chaperones. So there was an Anfinsen experiment, spontaneous folding. He showed that how folding is done. So there's a ribonuclease, is a small protein that contains 8 cysteine linked via 4 disulfide bonds. So there are 8 cysteines. They are linked together with the help of 4 disulfide bonds. And urea in presence of 2 mercaptothenol, fully denatured uh, ribonuclease. So whenever urea is present in the presence of 2 mercaptothenol, they fully denature the protein. But when urea and 2-metachloroethylene are removed, they are both removed, the protein spontaneously refolds and correct disulfide bonds are formed. And sequence alone determines the native conformations. So awards were given to the Nobel Prize winner. He was awarded as a Nobel Prize winner in 1972 for his experiments. So this is how the Anfinsen experiments looks like. So these are the disulfide bridges with the 72, 65, 58 and 110, 95, 40, 84, 26, 4 disulfide bridges. So whenever this is in the native state, that is catalytic active. But whenever we add urea, mercapthenol, so all these disulfide bridges are broken and you have free thiol groups. You can see this SH group, free thiol groups there. Because this is an unfolded state which is an inactive, and disulfide cross links reduce to yield cysteine residues in this case. And then further removal of urea and mercapthenol. If you remove this urea and mercapthenol, they came into the native catalytic active states. Disulfide crosslinks correctly reform again. So today's topic outcome so far is that you are able to understand the protein structural organizations. You are able to understand the attributes of primary, secondary, and tertiary structures. At the end of the course, you will understand the basic concept of protein stability through some well-established classical experiments. So these are the various references books: Principle of Proteomics, Introduction to Proteomics, Protein Engineering and Designs, Mass Spectrometry, and a textbook. So all these things are being arranged from these chapters. So that is the end of this topic. So I will share this with you on WhatsApp group just now. Sometimes it works, sometimes it's not. Protein stability and folding. So let's start with the next lecture. That is a topic of X-ray crystallography. So we will play some videos also with that and some presentations together. So maybe you have, I can check. So we are done with the topic. You can take your questions. GRLO is a type of chaperone. Yeah, it's a type of chaperone protein. Please explain lectures. Yeah, I already shared the lectures with you. Chaperone protein is the which help in the folding of your proteins. That's it. So you can see these slides later. Yes, I already shared on the WhatsApp group. Yesterday, did I share it with you also? The introduction part. Was it there? Or did I forget? I tend to forget to share things later. So I usually, whenever I share things with you while teaching, so I share at the same time because there are a lot of workshops going on at the moment. So it's become chaos. So did you have already the first from the yesterday's workshop presentations with you or not? Oh, I have a lot of ebooks actually, so I can share with you later. So ebooks for proteomics. So don't worry, I have a lot of engineering and designs, protein engineering design, protein folding kinetics, protein structure stability and folding, some good papers you can go through, biocore concentration analysis handbook, the experiment that we just discussed, and principles of proteomics. So I have a lot of books there, so I can share with you. Don't worry. Okay, if somebody doesn't have, I will share them again in case. Okay, shall I share the books also now? Pache, the Levinthal paradox just says that the energy, while making from simple amino acid primary structure to the folded protein, the energy get reduced like a paradox, like a cyclone. From the top, it is bigger, and then the lower, the energy becomes very less while doing so. That's something it says about it. So you can go through. I will share some review articles here, so which will be helpful for you. Some books and. So So I'm sharing all the proteomics books that I have with me. Can I do this sharing later, because I think there will be lagging of work. I might. It says that it will not be enough, but I have already started to share with you. Okay, let's start the next topic. It's already 9. So basics of protein crystallography and X-ray diffraction. So we will discuss about crystallography and diffraction, X-ray diffraction in this topic. So let's start with fresh start now. Hmm. So in this topic, you will be acquainted with the basic principle of X-rays and phenomena of diffraction and how this can be used to decipher the structure of a protein. You will get to know the depth knowledge of instrumentations and mechanistic of X-ray crystallography, how it is done, X-ray crystallography, and how things are being prevailed in this case. So why structure determinations helps in understanding it. So how proteins interact with other molecules. So we will understand about that. How they perform catalysis in the case of enzymes. How interaction of proteins with other molecules, including protein itself. And miscoding or misfolding of proteins associated with the disease. So how it is done. What is crystallography? It's a form of very high resolution microscopy, enable us to visualize protein structure. This is our understanding of protein function. What is the principle behind X-ray crystallography? It is based on the fact that X-rays are diffracted by crystals. So mainly check the structure of protein by throwing X-ray rays, diffuse X-rays to our structure of protein, and then it will diffracted. That crystal will be diffracted in different ways. The X-rays will diffracted in different regions. So according to that, we can predict the structure of our protein. So why X-rays, not others? Because light has 300 nanometer, electron has 10 nanometer. So light has visualization of individual cells, subcellular organelles. Electron has shapes and protein molecules, whereas X-rays has which is zero point 0.1 nanometer to 1 angstrom. And atomic details of proteins could be seen here. So X-rays in the order of atom diameter and bond length, allowing these to be individually resolved, and no lenses available to focus X-rays, and crystals act as a magnifier of the scattering X-rays in this case. So this is the how X-ray crystallography is done, the steps in structure and determination: protein purification, protein crystallization, data collection, structure solution, structure determination. Crystal enlarged view, diffraction patterns are here, and then electron density maps, then atomic models. So these are the various phases that you can go through refinement to check your crystallography. So X-ray crystallography step 1 is to check the protein purification. So what is protein purification? It is a series of processes intended to isolate one or few proteins from the complex mixture, usually cells or tissues. Just you have to get out your protein from the whole cells or from the tissues or whole organisms. So why protein structure purifications? It is a correct structures, interaction of the proteins. Around 5 to 10 milligram of pure soluble protein, and their purity should be around 95%. So here you can see protein being extracted on the gel, various concentrations. So this is the highest concentration of protein that we are looking for. Then in step 2, the protein crystallization. After extracting your protein, what you have to do is you have to crystallize your protein. So how you could do it? You can crystallize in a single unit by unimaginable weak and arranges a huge number of molecules in the same orientations. Scattered waves add up in phase, increases signals to a level which can be measured. This is often the rate limiting step in the straightforward structures, and especially for membrane proteins. So here you can see a molecule looks like this, and this is the unit cell, and the crystal would be all the basic unit cells together, joined together as a crystal of each basic molecule. Then comes the protein in the step 2. Uh, Crystal must be less than 1 millimeter. No crack should be there. No incursion should be there, such as air bubbles. So there should be nothing thing like should be there to improve while doing your things. And how you could do it? You do it with the hanging drop method. So what you do is 1 to 5 microliter of protein solution is suspended over the 1 milliliter of containing precipitant solution. So you take like hanging drop, this like a slide. You put up your protein on that, and there's a reservoir with the precipitants in it. And then you put that in the opposite side with the precipitant solution. Examples are ammonium sulfate solution or polyethylene glycol. So like this, you can have the crystal structure being formed in this bubble. So how X-ray crystallography in the step 2 it works on the basis of X-ray diffraction. So here you can see that there is a primary beam come and they click on our crystals. So like B, C, D, the primary beam came, diffracted, and they got into the diffracted beam. And they made these bonds, and where is your BC is equal to CD, and it's equal to the D of the diameter, the width of your diffraction, and is equal to sine theta, the angle of sine of this. So this is the main formula that they have diffracted, and they got the Nobel Prize winner for this also. Then we are done with the protein purification, then we are done with the crystal formation. Now comes the third step, that is data collection. So how the data collection is done? It is done with the help of mounting crystals. That is, crystals are mounted in a way so that samples can be rotated and an X-ray beam can be passed through the samples. So like this is the camera which will be taking the whole picture of your beam, and it will be revolving around it, and there will be one beam of X-ray, and here will be a sample. So methods of mounting include using either capillary or a tube. Both capillary and tubes are mounted on a goniometer. So exposing X-rays, once the crystals are corrected mounted, they are exposed to X-ray beams. X-ray sources includes synchrotron, giving high resolutions and luminosity, and X-ray generators for smaller laboratory use. So X-ray crystallography, step 3, data collection. The source of X-ray is often source of X-ray is synchrotrons. From the synchrotron, you can have the X-rays, and typical size of crystal for the data collection may be 0.03 by 0.032 by 0.01 millimeter. And the crystals are bombarded with the X-rays which are scattered from the planes of the crystal lattice. And the scattered X-rays are captured as a diffraction patterns on the detector, such as film on the electronic device. So here you can see that there's a rotating anode. It got focusing mirrors are there like this. So they're getting focused onto the crystals, and this is a Zeiss focus beams are there. Then they got diffracted, and you can check them at the area detector that we're looking for. Then rotate crystals through 1 degree and record XRD patterns. So you can calculate the XRD patterns of your crystal, and it's very crowded. Repeat 30 degrees were obtained in this case, and higher resolutions while using synchrotron. So this is the fourth thing, that is step is structure solution. to understand the phasing, that how your structures looks like from a diffraction data to electron density. A typical diffraction from a so this is the on the left hand side that you can see is a typical X-ray diffracted results from the first time, but with the help of electron density map of the left side, we can create a 3D structure out of it. That is on the right, density map of the crystals. So topic outcome for today is, in this case, is that students, after completing this lecture topic, you are able to understand X-rays and its applications in proteomics. You're able to understand the principles of X-ray crystallography of proteins, and you're able to theoretically overview how to elucidate the structures of unknown protein based on X-ray diffraction data. So these are the references books. So after this, let's play one video.
My work is geared towards understanding the mechanical connection between the nucleus and the cytoskeleton.
So I hope today's topic is much clear now with the extra crystallography with these two videos, and I will share them just now so that I do not forward later. And this the books already shared with you. Okay, books are shared. So let me see if I can share the videos also, because they are big, and big things WhatsApp does not allow to share. Let's be lucky today. So extra crystallography video I cannot share, but the rest was fine. Or this one is shared. Okay. Extra crystallography Bragg's law will be not shared, but other video has been shared, which was saying about the topic. So we are done for today, students. We are done. So if you have any questions, please feel free to ask. I want to start with the second topic also, but that will be too much for today. You will be not with full of energy, I guess now. And does Leventhal paradox and chaperones work on somewhat the same principle, or are they completely different? Cause the function seems to be same. I mean, chaperones are proteins, which are folding help in the folding of the proteins further. And Leventhal paradox is a yeah, it's a mechanism. So you can say it actually. So that's it for today, students. Will not take much of your time. So you can everything has been shared with you. Uh, Go for it, and yeah. So I will end the session now. Thank you very much for your time, and see you tomorrow. Wish you all good night. In German we say vielen Dank für heute, um, for being eine herzlich audience, what sagt, for being a nice audience and being nice students. Und ich wünsche dir viel Flow. I wish you all a lot of good luck and good life. Bitte, bitte. Gracias, gracias. Mercy. Deadline is 20th of October. So just be relaxed, take your time to submit it. Bis bald. So a lot of Germans are here. Bis später. Tschüss. फिर मिलेंगे. Namaste. Auf Wiedersehen. बच्चे bis morgen. Ja, okay. So we are done with the topic for today. So topics I will share by tomorrow morning. Sorry, I was traveling today. I couldn't share it. But if you have time, just start with your things then for the review paper. So I will share soon. If you have no further questions, I would like to end the session now and see you tomorrow, same time, same place with more learning. Thank you very much. Thank you so much. Bye bye. Good night.